Fantastic. The podium is yours.
Okay. Well, thank you very much. Is it in full screen mode?
No. Now, yes.
Yes. Okay. Great. Okay. Well, thank you very much for the invitation. I'm of course wish I could be there in person, but it's very nice to be able to participate remotely. And I hope all of you are doing as well as possible under the circumstances. It's been a very long period, of course, and actually I think the first lecture I gave during this, or first summer I gave during this entire lockdown was also involved at Hugo. So it's really nice to be back here again a year later. So today I'm going to talk about what's been really the dominant research program in my lab for about 20 years now. And that's looking at the evolution of morphological diversity across different groups of organisms, mainly vertebrates, and in fact, mainly mammals in my past, but now diversifying across all vertebrates and even dabbling in some of the insect groups lately. And I think we can all appreciate that understanding the evolution of diversity is really a fundamental topic in biology. It's central to, of course, appreciating and understanding how we got to the kinds of diversity that we see on Earth today and how that's changed in the past, but also for projecting how that's going to change going forwards as we go through a period of really unprecedented climate change and habitat degradation. And anybody who really thinks about biology or organismal biology on really large scales, the way I think many of us do, will already appreciate that diversity is not evenly distributed. So if we look across space, look space in terms of geography, we can see that there are many more species in the tropics than at the poles. If we look through deep time, we know there's classic patterns of periods where you have kind of high standing diversity or rapid increases in diversity, and other periods where you have rapid declines in diversity, as in the mass extinctions. And we also know that when you look across the tree of life, some groups of organisms have far more diversity than others. So you have groups like birds and fishes that have huge species numbers, and other groups like crocodiles or turtles that have relatively low species diversity in comparison. And so what I've really been interested in throughout my career is trying to understand what processes shape organismal diversity over these large scales. Now, one thing to point out is all those examples that I just gave you are focused on species diversity. But what I'm actually thinking about and talking about when I talk about diversity is the diversity of form or variation in form. And really, I tend to call this an organism's phenome. This is really the sum total of their observable physical characteristics and can include things like behavior and ecological aspects, and also ties them to their genomic or environmental drivers. And these characteristics are really important to look at, I think, because they mediate the interactions of an organism with other individuals, with other species, and with their environment. And from the perspective of a paleontologist like myself, it allows us to reconstruct their ecology and life history when we really have no other way to directly access that information. We can use geochemistry in some cases or taphonomy in some cases, but most of the information that we get about the ecology and life history of extinct organisms comes from their morphology. Now, this is of course important because this means that we can actually start to look at the evolution of form and the evolution of ecology and the evolution of life history in extinct organisms by pulling in data from the fossil record and building them into our comparative frameworks. And this is important because, of course, these extinct organisms don't just represent, mean shapes of the living diversity, but represent a huge increase in variation. In fact, it's really important to recognize that the vast majority of organismal variation is extinct. So it's not just that the majority of species are extinct, but the vast majority of variation across organisms is represented only by the fossil record. And indeed, when we look at just these kind of classic examples of some of the really weird things that have lived in the past, we can appreciate that they look very different from their modern descendants, for those of them that still have some modern descendants. So of course, the non-avian dinosaurs look quite different from modern dinosaurs, birds. And even if we look at things that went extinct relatively recently, like sloths or giant ground sloths, even though they are a relatively a recent group, and we have, of course, a few species alive today, the extinct forms look radically different from the ones that we're used to today. And so I think this really emphasizes that when we want to understand the factors that really shape diversity through time, we want to include the fossil record as much as possible. Now, it's not always possible. Certainly, most of the examples I'm going to talk about today don't actually incorporate fossils that much, because for some groups, the fossil record is either very poor or is quite deformed or flattened in some cases. We are working on reconstructing those sorts of specimens to pull them into our data sets. But for other groups, we do have well-preserved fossils. But even if we can't incorporate fossils directly into our analyses, we want to make sure that we think about that variation and keep it in mind, and make sure that any hypotheses that we come up with or any results that we generate make sense in light of the organismal variation that we aren't sampling, but that we know exists. Now, diversity or time isn't only deep, time is also developmental. And so one of the other aspects that I also try to keep in mind in thinking about the evolution of morphological diversity is the evolution of developmental systems. And this really follows from this nice quote by Conrad Waddington, that the evolution of organisms must really be regarded as the evolution of developmental systems. And I think if we just look at one obvious example, the axolotl, this is a salamander that looks extremely different from its closest living relative, which is kind of a tiger salamander type of thing. But if we, and we might, if we don't know anything about their development, infer some really huge changes. But if we know something about their development, as we do, we know that it's actually a fairly trivial change in developmental timing that creates these two very different adult morphotypes, or at least sexually active morphotypes. And so we want to keep this kind of information in mind as much as possible as well. Now, ideally, what we could do is quantify morphology throughout development and incorporate it directly into our models. But unfortunately, comparative ontogenic data is actually relatively rare, and actually maybe impossible to get for most of the living taxa in the world, but especially for fossil taxa, for which we won't have a lot of information on development. And so it's really thinking about how can we kind of access these sorts of information. We know that development is important for generating diversity. How can we access that if we don't have access to developmental data for the vast majority of life on the planet? And it's really thinking about that question that got me interested in the field of morphological integration. Now, morphological integration is a field that was really developed by Olson and Miller in the 50s, and it's based on a really simple idea. And that idea is that evolutionary changes in one trait do not occur independently of changes in other traits, or very rarely occur independently of changes in other traits. And by quantifying the relationships among traits, their correlations or their covariation, we can identify the integration of traits, the correlations among traits. And in doing that, we might look at something like, say, an entire skeleton and find that the entire organism is very highly integrated. Or we could find the opposite, that there's no correlation among parts. What we usually find when we quantify these relationships is something a bit different, which is that we have modules. And modules are groups of traits that are very, very highly integrated within themselves. So they have really high correlations amongst themselves, but relatively weak correlations between these groups and other groups. And so you have high integration within modules, but relatively weak integration between modules. And this seems like this is a pattern that we see in many different organisms, this pattern of modularity. Now, the idea of modularity is a really interesting one, and it's been looked at from lots of different angles over several decades now. But it's really grounded in the realm of genetics and development. And so a lot of the work that's been done in this field is really looking at in model organisms and model systems. And only in the last decade or two has it really kind of grown out into the macroevolutionary realm at a large scale. There's a lot of interesting things that we can tease apart within this realm of modularity, and things that are really relevant for thinking about the large scale evolution of diversity. And so I'm just going to point out a few of those things here. So one of the really interesting things about looking at modularity in phenotypes and integration in phenotype, is that a number of studies have shown that the integration or the correlations among phenotypic traits actually reflect the relationships among those traits in terms of genes and in terms of developmental pathways. So if you see highly correlated traits in terms of phenotype, you can infer that there's some relationship among those traits in terms of the genetic architecture or developmental patterning of those traits. Now, a lot of times we might tie these relationships to function, so suggesting that certain traits are functionally aligned, and that is true. And Young and Hogginson made this point, I think, very clearly in one of their papers, where they stated that the functional association among traits may drive selection for integration, but the underlying target of that selection is development. To produce high levels of covariation, essentially to have, that being heritable, selection must be acting on the underlying developmental architecture of traits. So again, that means that when you're looking at a phenotype and when you're looking at the relationships among phenotypic traits, what you're actually able to access is the underlying developmental and genetic architecture of those traits. So that's one of the really interesting points here, again, from the perspective of a paleontologist that rarely has access to direct developmental data. The other really interesting thing I find about modularity integration is ideas about how they themselves evolve. And so this is something that a lot of people have speculated on or hypothesized about. And essentially the idea is that, that essentially modularity has increased over the course of organismal evolution. So that ancestrally, we see more integrated phenotypes. And as evolution proceeds, as developmental pathways get more complex, you see an increase in modularity, where you get a fragmentation or a breakdown of the relationships between modules, maybe as things become less functionally related. Now, the opposite is also entirely possible, where over the course of evolutionary time, different modules become integrated. So you can see fragmentation or integration when you try to quantify these patterns. So that's one interesting and completely open question is there any trend towards increased modularity or increased integration through time? And the other interesting point there is why? Why would you see these patterns? What difference does it make? And that, of course, to me is the most important and most interesting thing about modularity. The first one being that it preserves the imprint of these genetic and developmental and functional associations just in the phenotypic data. The second is that those relationships among traits are a primary influence on morphological variation and the evolution of individual characters. And the way this would work is that if traits are highly integrated with each other and one changes in a certain way, then the other ones have to evolve in a correlated fashion. Now that may negatively impact any of their functions. And so the idea is that high integration of traits may constrain the evolution of any individual trait. And that modularity evolves as a way to break those functional relationships so that different traits can evolve independently from each other. And that potentially increases disparity, increases variation, and may be related to evolvability. So that's a really interesting question, because then the evolution of modularity itself is fundamental to the evolution of diversity. Now, again, these are hypotheses, and they're difficult to test with empirical data. But there's been a lot of work on simulating these effects, and then some empirical work too. So I'll talk through both of these. First, I'm going to start off with some fairly simple simulations just to break down those patterns a little bit more clearly. So the idea here is that the relationships among traits are limiting the variance of what the traits would be able to do if they were evolving entirely independently. You could phrase this as suggesting that these relationships are limiting variance to specific regions of morphospace. So we can simulate this, and to be honest, this is actually mathematically quite obvious, but I think it's still helpful to look at personally. So in this plot on the left here, this one that kind of looks like Saturn, what this is a plot of just each of these little dots represents a phenotype, and I just let them evolve, under a random walk model. And the red ones are evolving with no covariance among the traits, so no integration among the traits. They can just do whatever they want. And as expected, they just diffuse through the morphospace in a fairly even pattern. Now, the black traits have integration. And in fact, what I imposed on them was a kind of standard mammal skull covariance structure. Now, as you might expect, what happens is that those traits The evolution of those traits falls along a few axes of variation. Those are the principal axes, which are defined by the covariance structure of the matrix. But what's interesting here is a few things that becomes very obvious, which is that huge regions of the morphospace are not explored because of the integration among traits. But the other thing that's interesting is by forcing all the variation into just a few directions of variation, you actually go further along those limited trajectories than you would normally expect given just an entirely free random walk model. And so these areas, I don't know if you can see my mouse, but these areas at the far ends of the space here represent regions that may be actually being facilitated by the integration of traits as opposed to an entirely constrained pattern. So you may actually expect both more and less evolution in different directions because of integration. One of the other things to point out here is that by pushing all the variance into just a few directions, you could also expect to see much more convergence because there are fewer solutions to problems, because they can only move around in a kind of certain amount of morphospace based on their covariance structure. So that's an important point, I think, because we often treat convergences and in shape among different groups as a really unexpected thing. But I think actually, if organisms have a fairly similar developmental pathway or developmental patterning, that means that they have similar developmental architecture, similar covariance structures of the traits, then actually we probably would expect convergence as the expectation, as opposed to a sort of unusual phenomenon. And that also relates to homoplasia as well. So integration, you can expect, will increase homoplasia, and is of course then problematic for phylogenetic analyses. So we can also flip this on its side and look at it from a little bit of a different angle, which is what this figure on the right is about. Now this figure on the right, time is on the x-axis, and on the y-axis, we have divergence from the ancestral phenotype. Again, these are simulated traits. So we're starting off from some 0-point ancestral state and then simulating these shapes. And again, the red ones are the ones that have no correlations among the traits. They can do whatever they want. And the black lineages are ones that have integration of the traits. And what we can see is that the ones that stay the closest to the bottom all the way along time, the ones that basically diverge the least from the ancestral phenotype, are the very integrated traits. So these are the very integrated phenotypes. So these are the ones that are staying really in that kind of center area of morphospace and are being constrained from evolving very much. But if we look at the very top of that figure, we also see that the phenotypes that are diverging the most from the ancestral form are also highly integrated. So again, you get both more and less evolution with integrated traits than you may expect from an entirely unintegrated phenotype. So that's quite interesting, because that means that really, you can make either argument, and we don't know which one actually dominates in the natural world, but either one really could. I just also want to point out that here I'm really talking about variance or disparity. It's not so clear how this translates to evolutionary rates, actually. And in fact, what we have found in some of our empirical examples is that we find parts of the skull, for example, in mammals that have really a high integration. So things like the basicranium of carnivorans, which is often thought of as a very conservative part of the skull, really good for phylogenetic data and things like this. It's very highly integrated. So the correlations among the traits in that part of the skull are really, really high. It has quite low disparity relative to the other parts of the skull, which fits this model of high integration kind of constraining disparity. But actually, when we look at the rates of evolution, we find it has actually one of the highest rates of evolution of any part of the skull. And so what that suggests to us is a model that's a little bit different. And this is a model that we've suggested that we call the fly in the tube model of evolution. And the idea here is that the covariances of the traits, or the integration of the traits, is kind of creating an area of morphospace of preferred shape change defined by the covariances of traits. But within that area, actually the fly or the phenotypes can zip around fast or slow exploring that space. It might stay in the same area and just buzz around really quickly. It may slowly meander through the entire thing. It can do all kinds of different things while exploring that space. In the case of the carnivorous basicranium, it seems to be moving quite quickly or evolving quite quickly, but in a very, very small region of space, so just re-evolving the same shapes over and over and over again. And so that's why I think the relationship between integration and rate is a little bit less clear than the hypotheses for how integration can influence disparity. So what these simulations leave us with are a lot of questions. We have two completely opposite extremes of what we might expect for integration and modularity and how they might impact macroevolution of form. And so we really want to figure out what effect dominates in the natural world. How do these trait interactions really influence the tempo and mode of phenotypic evolution? And also, I think it'll be really interesting to try and understand whether these trait interactions or trait integration have different patterns depending on ecological mode or life history mode. So are some of these different modes more evolvable, or do you see more or less integration in taxa based on whether they're metamorphic or direct developing or herbivores or carnivores, things like that. And then also, of course, the big question, which I definitely do not have an answer for today, is whether there is an evolutionary trend for increasing modularity through time to really circumvent the constraints due to trait integration and the potential for the integration of traits to really constrain the evolution of diversity. And all of this is really part of a kind of broader question, right? Which is, what are the intrinsic and extrinsic factors that influence tempo and mode of evolution of organismal form? And I think by quantifying trait interactions, we can get at this intrinsic aspect of it. And then we have many other ways of looking at the extrinsic factors, methods that are really more common in the world of paleontology and macroevolution. By quantifying these intrinsic patterns of integration, we can put all of them into a single framework and try and look at how all these different factors contribute to the evolution of diversity. Okay, but so in order to do that, what do we need to do? We basically need to quantify and compare morphological evolution and trait integration across taxa. And people have done this, mainly in mammals. Mammals are certainly the most studied group when it comes to macroevolution of trait integration, specifically in the skull. And certainly at this point for about 3 decades now, people have been working on this in mammals. And when you look at mammals, the overall pattern is generally very highly conserved. Depending on what kind of hypotheses you test, there's about, say, 6 cranial modules that seem to be identified in the cranium. And there's really fairly few exceptions. Monotremes, the platypus, this would be one exception. We tend to find that some of the very aquatic mammals like whales or seals also seem to show some deviations from that pattern. I think there may also be a pattern shift in the anthropoid primates, which is something that we're just writing up now. But by and large, mammals are pretty conservative in their pattern of cranial integration. And that means that gives us relatively little capacity to look at what drives changes in trait modularity and integration, and what the evolutionary outcomes of differences in those patterns actually are. So we need more variation in trait integration in order to understand what the actual consequences of trait integration are for evolution. So what that means is we need to actually quantify and compare morphological evolution and integration across a broader range of taxa. And so that's what we've been doing for the last several years. So essentially with my lab, we've been undertaking a project to really travel around the world and build a huge 3D data set of skulls for all tetrapods, or spanning all tetrapods. So these are all the land vertebrates. And so far we've collected about 2,600 specimens, which represents about 1,900 species. And about a quarter of those are fossils. So these are really nice, undeformed, 3D preserved fossils. Now those are definitely concentrated in certain groups. Things like mammals have really fantastic fossil records. Things like birds, not so much. Well, we put together a really nice representative data set really spanning the diversity of this group and its fossil record as much as possible. And the reason for the discrepancy between specimens and species is because within each of the major clades, we have some species that we have really large intraspecific samples for, so 40 specimens or so per species. And then we can look at the patterns within species and compare them to the patterns that we find across species to really look for the conservation of patterns of integration at different evolutionary scales. And that seemed like the hard part because we spent, obviously a very long time traveling around collecting all of those scans. But once we had those scans, we came up with a far more difficult problem, which is how do we actually quantify and compare all this diversity? And our goal here, of course, is to have an accurate, comparable, and really a high resolution description of phenotype. And that's important because the first thing that we did here is to take all of our different groups. So, I was working on the mammals, we had somebody working on squamates, Aki was working on squamates, Ryan was working on birds, Carla was working on the amphibians. We came up with a list of landmarks for each of our different groups and then compare them across our groups. These are your normal type 1, type 2 homologous landmarks. And across the entirety of our sample, we could only identify about 12 homologous landmarks that we could pick out consistently. And when you have these amazing 3D skulls, having just 12 landmarks to really describe their shape feels very unsatisfying. And that's, of course, because if you just look at these images in front of you, a huge amount of that diversity is entirely left out when you're only using a few landmarks. So a lot of these very weird processes, certain bones that aren't present at all in some taxa, but are hugely significant in other taxa. All kinds of different structures are going to be entirely left out when you're only picking out 12 landmarks. And so we went a different route. I should say that trying to figure out the solution to this problem felt very much like Sarlacc's Pit for you Star Wars fans out there. It was a really, really difficult process and it took a very, very long time to figure out. Not quite 1,000 years, but definitely a few. But eventually what we came up with was to really apply a surface morphometric approach. And so what we do here is we take our 3D scans, clean them up, and then we apply our discrete kind of type 1, type 2 homologous landmarks onto all of our groups. And that will be different for each group. We will have our kind of 12 common ones, but also the ones that are more specific to each clade. So mammals will have a different set than frogs and things like that. And then we use curve semi-landmarks to link up our landmarks and really partition out the different regions of the skull. So it looks something like this, where the yellow landmarks here are curves, and they're really partitioning out like the maxilla or the jugal, squamosal, etc. So the different elements, in some cases, it might be a combination of elements if there's ones that can't be separated. And then we take that and we apply that same those same landmarks and curved semi-landmarks to a really generic template. And then on that template, we place a series of surface semi-landmarks, which are specific for each region of the skull. And so those semi-landmarks, those surface points, are common across our data set, across our tetrapod data set. And then what we use is code in R in the morpho package to automate the application of those surface landmarks to our specimens. And so just to see like that last step there, what this essentially does is it takes this generic hemisphere, it warps it to the shape of each individual specimen, it projects the surface points from the template onto the individual specimen. And then what we have for each specimen is the same distribution of surface semi-landmarks, which is guided by those initial landmarks and curved semi-landmarks. And what that gives us in the end is a really nice data set that's comparable across all of our different specimens. Now, there's a little bit of variation here because these are actually clade-specific data sets, but then we redo them so that with a common scheme for all of them, which is a little bit less well sampled because there are obviously huge differences between some of these groups. But we have a data set that is comparable now across the entire breadth of tetrapods. So we can think about frogs and think about whales and think about patterns in crocodiles and know that we're comparing like for like when we're thinking about, okay, what is the pattern of how ecologies influence the shape of the skull in this group versus this group. We know that the differences or similarities that we see aren't just because of the kind of data that we're collecting, they're actually recording biological differences or similarities. Now, that was hard, but actually what we also found out is that once we have those data, we need to analyze them, and that creates a series of new issues because of course we're looking at these data sets both in terms of specimen numbers, species numbers, and landmark data at a scale that other people have not done before. And that just requires a level of computational power that that is really just a new scale. So we had to do a lot of testing of methods, a lot of trial and error, and a lot of development of new methods to actually be able to handle large multidimensional data sets, because the vast majority of methods that people put out there for evolutionary modeling are either entirely univariate, or if they are multivariate, they've only been tested up to say 8 or 10 landmarks or variables. And so that's required a lot of work, especially in collaboration with Julien Clavel, who's down in Lyon, who's really doing some of the best work, I think, in this field. And of course, one of the other things we wanted to do is make sure this was actually worth it. Are we really getting much out of these additional data points that we're adding, or are we really just replicating the pattern that we would have gotten with our landmark data anyway? And so we did do a series of subsampling analyses to look at differences in our results if we were only to look at landmarks versus our entire surface landmark data set. And a lot of the patterns, the overall kind of big picture story is fairly similar. It was pretty similar. But actually, if you really want to look at the nuances of the shape of the skull, landmarks will give you about half of that detail in terms of the description of skull shape. And so what we found is that for each skull bone, looking at Sicilians and squamates as our test data sets here, you probably need to say 15 to 20, 25, depending, landmarks and semi-landmarks per each region to really capture essentially the full shape of that structure. And that's something that's important because we probably are oversampling by about double. So it's good to know that we don't really need that much data. And another thing that was interesting is that actually the vast majority of that information is captured in those curves. Now the surfaces add a lot of important information, especially if you have things like the tentacular foramen in things like Sicilians, or if you have lots of cranial ornamentation, the surfaces give you a lot of information that curves won't give you. But for a lot of taxa, the curves are giving you a ton of important information. And once you have all that done and have solved all those problems, you can start doing something that we just couldn't have done before, quite frankly, which is starting to look at very, very different taxa and trying to pick out the similarities across them. So here we have, a theropod dinosaur and a Sicilian amphibian, two extremely different kinds of animals, but with similar data for the different regions of the skull, even though they have different bones in a lot of their skull. We can start to pick out things like identifying that certain regions of the skull are consistently highly integrated. So one of the things that we keep finding is that the suspensorium, things associated with the jaw joint region, whether it's one bone or several bones, are consistently very highly integrated. The occipital region is also consistently very highly integrated. The facial bones actually tend to vary quite a lot across the different clades in how they're organized. So in some groups they're more integrated, other groups are less integrated, and the same bones aren't necessarily integrated in the same way across taxa. So we have parts of the skull that have a really common pattern across all tetrapods, and other parts of the skull that are more variable. And again, it's a kind of comparison that you just can't make unless you have the same kind of data for all of your taxa. And so once we have all these data for all of our different groups, and we have them, because we're doing this surfacing bone by bone in order to be able to look at patterns of modularity and being able to look at the different parts of the skull separately, as opposed to a kind of pseudo landmark approach, which would give you the entire shape of the skull, but not the individual regional information, then we can do lots of exciting stuff. And so what we've been doing, obviously, is for each of these groups, going back to that original question of what intrinsic and extrinsic factors really influence the tempo and mode of evolution of organismal form. And so I'm just going to give you a really quick overview of some of them and then pick out a few to talk about in a little bit more detail. So for example, we've looked at snakes and lizards. We have a paper in review right now looking at crocodiles. We've had a couple of papers on birds and dinosaurs. Recent paper looking at salamanders and frogs. And then we're really right now working on the mammal data sets and wrapping those ones up. And for all of these, we've used our understanding of the relationships among taxa to reconstruct how the shape has evolved, looking at how rates of evolution have changed through time or changed across the tree, trying to look at what kinds of factors are associated with fast or slow rates of evolution or with high or low integration. And looking at not just what's driving shape, but whether shape, like the variation that's really driving shape is also driving rates of evolution, or whether those are disconnected. So I'd love to talk about every single one of these papers in detail, but I'm just going to pick out a few of these at the top, the dinosaur, bird, dinosaur, and salamander papers, to just give you some of the highlights. And so the bird and dinosaur work is really led by Ryan Felice, who's a lecturer at UCL now. And one of the things in this data set that we really wanted to look at was this question that's really goes all the way back to Darwin, of course, about whether diet and skull shape are really strongly associated. It feels like they should be, and yet time after time, analyses of large-scale analyses have shown that actually there's not a strong correlation. So we thought with our better, more expansive, better sampled data set, we would find that that was the case. But in fact, it's not the case. Shape of the skull or shape of the rostrum is not significantly associated with diet across birds. But interestingly, the rate of evolution is significantly associated with diet, in that seed-eating birds and nectar-eating birds show faster rates of evolution in the skull compared to other kinds of dietary groups. And that's of course interesting because this classic idea of these adaptive radiations in birds, all of the groups that people talk about are in fact seed-eating birds and nectar-eating birds overwhelmingly. And so it makes you think that if Darwin had looked at, carnivorous birds or something like that, maybe we wouldn't be here today. Well, we'd be here, but we wouldn't be talking about this. Okay, so that's one interesting thing that came out of that analysis. But we also, of course, wanted to look at modularity and rates of evolution in the different parts of the skull. And so what we did here was look at the correlations of the different cranial regions, identifying what the pattern of modularity was. So we found that the bird skull is quite modular with 7 distinct regions. But actually, those regions are quite dissimilar in terms of their degree of integration and their rates of evolution. And in fact, we found that the parts of the skull that have quite complex developmental histories, in that there's multiple tissue types contributing to the structures, actually show both low integration and fast rates of evolution and high disparity generally. And these are also ones that incorporate the anterior mandibular cranial neural crest tissues, as opposed to ones certainly that have mesoderm, which are kind of consistently high integration, slow evolving. Now that's a hypothesis, of course, and what we're trying to do now is actually do some experimental genetic work to really test that hypothesis. But it's an interesting hypothesis for why we might have differences in the rates of evolution of different parts of the skull, that it reflects both tissue origin and complexity. Now, well, the other thing we can do with these data, with these nice high resolution data, is try and estimate the ancestral morphologies. And so here's our estimate of the ancestral bird. And what we're doing now with a new PhD student in my group, Eloise Hunt, is actually reconstructing flattened bird fossils in 3 dimensions. And then we're going to build them into this data set and really kind of test that hypothesis more robustly. Now, of course, birds do have some good fossils, especially if we think about stem birds, also known as dinosaurs. And so we took our bird data set and we expanded it out to non-avian dinosaurs. And again, using that same kind of high density surface scheme, which we of course had to modify a little bit to deal with parts of the skull that weren't very well preserved, like the palate, and also just different regions of the skull that had to be slightly combined for the dinosaurs. And when we did our principal components analysis, which you see in the top halves here of this figure, or of this slide, is that the black dots are the birds, and the colored symbols are all different kinds of non-avian dinosaurs, or stem birds. And what you see is, sure, we have obviously a ton of birds in our data set and relatively fewer non-avian theropods or non-avian dinosaurs. But the non-avian dinosaurs actually cover a huge range of the morphospace in this figure. And actually, when we reconstructed the rates of evolution across this data set, and these are the figures across the bottom for the different regions of the skull, what we found is that consistently, the non-avian dinosaurs had the highest rates of evolution for the skull. And so these trees down here, the fastest rates of evolution are in red, the slowest ones are in blue and green. And you can see that in general, the non-avian dinosaurs, which are sort of more towards the center of the plot, sort of at 2 to 3 o'clock position, have the hottest rates of evolution in most of the cranial regions. And we did lots of kind of testing of, branch lengths and sampling and all sorts of stuff to really see if this pattern was robust, and it overwhelmingly was. And when we look at all the different parts of the skull, in this case, the first part of each of these little bar graphs here is the birds, and then we have non-avian theropods and then non-theropod dinosaurs, breaking those two groups up a bit. And consistently, we find that the birds are the slowest evolving in every part of the skull. And so this really reflects, that birds are great. Birds have huge radiations and are very interesting, but compared to other dinosaurs, birds are not doing that amazingly in terms of exploration of morphospace and how quickly they're evolving really different forms. And this actually, if you think about non-avian dinosaurs, they are hugely diverse in skull shape, and they're also doing a lot more with their skulls than birds do. They are doing things like really diverse feeding mechanisms. They're doing things like signaling and fighting. They have much more complexity of jaw musculature compared to birds. And what this really likely reflects is a shift in what the skull is being used for in non-avian dinosaurs, where it's doing both lots of kinds of different kinds of food acquisition and processing, as well as ornamentation and signaling and fighting, and other things like really complex sun production, to birds where the skull is really focused on brain protection and basically has a beak stuck onto the front of it. And in fact, most of the signaling and things like that happens with the hard or the bony parts of the skull in non-avian dinosaurs, birds are doing with their feathers instead, again, maybe driven by needing to be light and again, protect that brain. So what we see is a clear downshift in evolutionary rates when we get to birds, and a quite different kind of mode of evolution compared to what we see in non-avian dinosaurs. We also see a shift in what parts of the skull are evolving. Really within birds, really it's the rostrum is where we see the fastest rates of evolution, whereas in non-avian dinosaurs, we see a lot more evolution going on around the jaw joint and around the back of the skull. And so we can see, these differences in which regions of the skull are really diversifying the most. So I'm just going to briefly switch to the other group that I think we've seen really interesting results for over this past year, and which I think are arguably the most interesting vertebrates, which are the amphibians, and especially some work on salamanders by Anne-Claire Fabre. And again, using the same kind of paradigm of high surface density, high density surface landmarking of the skull, Anne-Claire did this really lovely study looking at what factors are driving skull shape evolution in salamanders, and found that overwhelmingly we find that skull shape reflects how they're developing, whether they're pedomorphic, biphasic, or direct developing. And so in this principal components analysis here, you can see the different color symbols reflect their life cycle type, and that is the far more dominant pattern here compared to things like habitat or other ecological modes. And when we reconstruct rates of evolution across this entire group, we also find that you see shifts in the rate of evolution associated with shifts in life cycle. So if you go from biphasic to pedomorphic or direct developing to biphasic or things like that, you tend to find a shift in the rate of evolution. And in fact, it's the direct developers seem to be the slowest evolving, while the biphasic and pedomorphic forms seem to be the fastest evolving. Now, this is really interesting, actually, because we just recently, a month or so ago, published our paper looking at the same kind of analyses in frogs. And we actually find the opposite results, where frogs show much stronger signal of habitat on skull shape and also on rates of evolution. But actually within frogs, within on the side of reproductive strategy, although it doesn't have a strong influence on the skull shape, we do find that every time that frogs lose their larval feeding mode, so they don't have a feeding stage as tadpoles, they don't actively feed as tadpoles, they actually show a burst of evolution. They evolve faster once they lose that actively feeding larval stage. So actually quite a different pattern than what we really see in salamanders, both in terms of having a stronger influence of habitat, but also this faster evolution in the direct developers compared to the more biphasic forms. Not what we would have expected. And then finally in the salamanders, we also find that the ones that don't go through any sort of metamorphosis at all, the paedomorphic ones, show the most integrated skulls compared to the biphasic ones, and also the direct developers, which actually do go through metamorphosis, but in the egg. So no metamorphosis seems to be associated with higher integration of the skull, which is interesting to consider. And then finally, I just want to close by pointing out that now that we have these really kind of unified data sets across this diversity of forms, we can really start to understand what are the different factors that drive skull evolution and identify the commonalities and differences and start to interrogate those in more detail with things like experimental work in some of those systems. So hopefully I've convinced you that this approach is really powerful for understanding morphological evolution across really disparate groups. And that trait integration is a really big piece of this puzzle because it can significantly influence morphological evolution by limiting evolution to limited areas of morphospace, but also facilitating change across lines of least resistance. So you can get more trait evolution than you might have otherwise expected. Vertebrate skulls are highly modular, but not all in the same way, although there are some really consistent regions like a highly integrated suspensorium and occiput, and both development and function, including things like reproductive strategies or habitat, are really major influences on phenotypic evolution and integration. But there's a lot of variation in how important they are across different tetrapod clades in ways that we wouldn't have expected. So we're just finishing up the last parts of this data set, and then we're going to do a really big combined analysis across all of them. So stay tuned for that. And we're also doing things like continuing our methods development to be able to deal with data sets like this, build in things like climate into our models. We're expanding to systems such as arthropods and pelagic fishes, developing new methods to automate some of those landmarking tools, and again, interrogating the mechanisms underlying those patterns by bringing in genetic and developmental data more explicitly. So if any of those are projects that any of you are interested in, if you're looking for postdocs, do drop me a line. We have a really great track record of fellowships at the museum, and I'd love to talk to any of you who'd be interested in trying these sorts of approaches with really any kind of clade or data set. So do drop me a line. I'm always happy to talk to people about opportunities to come work with us here in London. And so let me just close by thanking my absolutely fantastic lab who figures out how to do all this really, really hard stuff. Our funders who make it, of course, possible to do it, and all the people at institutions around the world that give us access to all of these collections. And as we publish these data sets, we put them all online on Phenome 10K for free download so that you can use them for your own work. Thank you.
Thank you very much, Anjali. Wow, was an amazing talk. Always impressive with all the figures. They're fantastic. All the scan and the pretty of all your figures, always a pleasure to see. So now I will open the session of questions. So everybody who wants to ask any question can raise the hands using down. También para las personas que quieran hacer preguntas en español, podemos también aportar en ayudar en las preguntas, la traducción, levantando la mano. Ahí hay una pregunta. Voy a permitir que se quiten el audio. Un segundo, por favor. Bien, Raúl, puedes preguntarnos.
Hi, thank you. Anjali, many thanks for your talk. It was really inspiring. I'm wondering if you have in mind or you're planning to integrate your data sets with genomic data, since like there is many genomic data available for many vertebrates now. And I think it will be really interesting to compare the patterns of evolutions that you are finding with the pattern of genomic variation.
100%, absolutely. I think that's really, really interesting. So there is a postdoc that's starting at the museum who is going to be looking at things like ultra-conserved elements and cranial modules with some of this data set. We're also collaborating with people in the BIRD 10K project who are putting together a huge comparative genomic data set. And so they're actually, so in that case, we actually talked early enough that we were able to align our sampling. So we've sampled the same bird species as they're sampling, as they've sampled. And so right now, actually, they're running the analyses to do exactly what you suggested. We have a little bit of work looking within, where we're starting to talk about some work looking at other aspects also within some model organisms and things like that. So actually looking at things like transcriptomics. But I think there's massive scope to do that still, and it's definitely a huge part of what I plan for the next steps of this. So yeah, it would be really great to, if that's something that you're interested in, definitely would be.
Awesome, thank you.
Okay, Matthias.
Yes, thank you for the very nice talk. And I have a question. So right now, all this multivariate data or analysis to analyze modular data is applied to morphology. And I was wondering if you see or how you see this kind of analysis applied to other kind of trait data, like behavioral, for example, if we can get some sort of multivariate modular behavioral data to include into this analysis.
Oh, absolutely. I think we absolutely can do it with, with really any kind of data that can be quantified or discretized in some way. You can apply in the same sorts of approaches. And so certainly, so one thing that we do is we of course take the trait data for things like life history state, or behavior is a little bit tricky because it requires actually having data on behavior, which unfortunately usually doesn't have as good of a sample as other things, although it'd be really great to have that kind of data, and we can reconstruct the evolution and ancestral states for those kinds of data and then directly integrate them into our analyses. For example, by using the sim tree instead of our original tree, to see how those different traits are related to the evolution of morphology. So yeah, it's completely possible, and I think that's a really, really critical thing to do, to see whether changes in behavior or other aspects other than morphology are also related to bursts of evolution, whether taxonomic or morphological diversity or ecological diversity, in fact. So yeah, absolutely. And there are methods to do that, certainly on the univariate side and increasingly on the multivariate side. So the real difference between how you would treat that kind of data versus morphometric data just relies on essentially the distribution of variance in those kinds of data sets, which depends on how they're being coded or scored and or quantified. But it's entirely possible to do, and a really good thing to get into.
That's amazing. Well, there's more questions now. Mahendrian.
Yeah.
Perfect.
Yeah, Anjali, it was very nice talk.
Thank you.
And my question is this: it's quite clear these days that modularity is happening at the skull level, and as you said, behavior also has enough role to play to make the modularity to take up to a level. And along the line, do you think that thinking would also be work in a modularity fashion in brains?
Oh, so do you think thinking happens in a modular fashion, or does brain evolution happen in a modular fashion? Which one do you mean?
Yeah, both the way. Explain both the way. What do you think?
So I think brain evolution definitely happens in a modular fashion. Jeroen Smers has been working on this a bit. He's based at Stony Brook, and so he's been doing a lot of work putting together different kinds of data sets for different how the different brain regions are evolving and the modularity of brain region evolution. And I think absolutely brain evolution is happening in a quite modular way. So yeah, definitely. And in fact, that raises the point that, you know, most of the work that people have been doing in modularity, certainly in vertebrates, is very focused on hard tissues because it's easier to quantify and things like that. But there's tons of interesting work to be done looking at soft tissue evolution and modularity of soft tissues, the integration of soft tissue evolution with hard tissue evolution. Those are things that are really tricky to quantify because they're going to be more affected by, you know, position and preservation. But I think, you know, we have to start getting better at doing that kind of thing. So that's one thing in terms of brain evolution. Now, is the evolution of thinking modular, or is thinking modular? I'm sure it is. I bet if you were doing kind of what functional MRIs and things like that, you could actually identify modularity. It's, you know, far outside of my field, but I can totally imagine that there is modularity in kind of, you know, brain activity. I don't know that anybody's looked at it quite that way, just because I don't know that literature, but it would certainly make sense as a topic to look at. Yeah, I wouldn't be surprised at all, given that, you know, the brain regions are quite modular. Why wouldn't their function be?
Probably that'd be true. Thanks a lot. Thank you.
Interesting question.
Definitely.
I'll think about it in a likely modular way.
There's two more questions. Also, I have one, but I will wait to be the last one. Nicolas.
Yes, thank you for your talk. Yeah, I had a question about so most of your experiments use some mostly like morphological integration, but I think that at least you show, but how could you be able to define some new models with this technique, and how could you test this model later, like seeing when this model assemble or disassemble during evolution?
Yeah.
How this compare with the information from ontogeny, from development works, like this kind of experiment where you try to manipulate the model and see which other part are affected?
Yeah, good question. So in terms of, you know, the approaches that we have for identifying the modules are, you know, model testing methods. So they are largely, the hypotheses are based either on functional or developmental hypotheses of what different components would be integrated. And so some of those, you know, are things like, you know, tissue origin, or can be based on experimental work, although that hasn't necessarily had such, I'm trying to think of where that's actually been explicitly tested, and I don't think there's been that much along those lines. One of the issues with it is that they can pick out some parts of it, right? You can look at whether specific aspects are modular that aren't necessarily incorporating anything in the entire, everything in the entire specimen. But one of the issues with this is that, as Hallgrimsson, I think, laid out in this really nice paper in the late 2000s with the palimpsest model, is that it's actually really hard, starting from end phenotype, to reconstruct the exact developmental aspects that are driving the patterns of phenotypic integration, because you get essentially, he called it the palimpsest model. It's sort of these manuscripts that people would write on and write over them and write over them again, because every, you know, next developmental patterning overlay is the one before it. And so what you end up with is sort of this mishmash of layers of developmental patterning. So it's really hard to tease apart those effects. And a lot of times the same, different pathways might produce the same expected pattern of modularity. So in that sense, I think starting, you know, we have a pattern now that we have identified, but in order to really tie it to the developmental pathways, we have to go into the experimental systems, I think, and actually manipulate, you know, individual pathways and look at whether the effects are limited to a single a module, or whether there are more global effects. And I know just from a very, you know, just from a entirely a qualitative perspective, you know, looking at some of the genes that have been identified that affect things like jaw development and things like that, you know, I have taken a look and, you know, to see, okay, well, are they doing anything in the basic cranium really? Does it look very different? But what we are trying to do next, and in fact, you know, I've been working on a grant to do exactly this recently, is to actually go into those systems and pick out the genes that we think, well, one using comparative genomics and transcriptomics to identify genes that may be differentiating groups that have different patterns of cranial modularity, and then experimentally manipulating them to try and build, test those hypotheses, and then we can build that back into our model. So it'll be an iterative process for sure. But right now, I think I kind of feel like we know a little bit too little, actually, to do that very well yet. But we're really close, I think, to the point where we can do that now with experimental systems, since now we actually know what we're looking for in terms of these differences among big clades, as opposed to just an entirely exploratory analysis. Now we can target it on these specific regions of the skull. And just to expand on that a little bit, one of the other issues I think in this whole thing is that we have these big differences across the major clades. So frogs, amphibians actually are pretty good. They're pretty similar, but we have kind of big differences between amphibians and amniotes. We have big differences between synapsids and diapsids and things like that. And we don't actually, because of sampling, we don't actually really have the capacity to capture the transition between those patterns of modularity. So that's one thing that is frustrating to me right now. And so I think the only way we're going to be able to do that is actually by manipulating things developmentally to try and see where those transitions are happening, because I'm not convinced that we're going to be able to do it with a fossil record given sampling issues.
That does sound hard.
Yeah.
That's definitely ambitious, but well, a very nice project for the future. Anjali, there's one more question. There's Monica Angulo.
Hi, thank you very much, Dr. Goswami, for the talk. I have a question about, well, I'm assuming that this study was done using like adult skulls. So is it possible to do it, I guess, with not this amount of species, but with a lot of species, like doing the ontogenetic integration?
Oh yeah, absolutely. So I've done this a little bit. Other people have done it more than me. But you can definitely look at this through ontogeny. So one of the things that I've done is look at like multiple stages going through mammal skull development, for example. Other people have done this in fishes and other groups also, or through various. I think there's been some work I think in Anolis also looking through ontogenic stages. And actually, Claire Fabre, who's a postdoc who led the salamander project, one of her plans is to actually start looking in more detail in the ontogeny of amphibian integration. So we have a little bit of data for mammals. We have a little bit of data for frogs already, and she's going to dive more into that with salamanders. So you can absolutely do that. One thing to keep in mind is that one of the difficulties with looking at ontogenetic integration is that you need to make sure that things are essentially at the same stage when you're sampling them, to make sure that you're not just looking at variation in level of development as opposed to integration at any one stage. But what is really interesting, and I think Miriam Zeldich really did, the kind of critical work on this in the 80s and 90s, is that and then I think all the subsequent work I've ever seen has really, well, not all of it, but a lot of it has confirmed this, is that integration is actually really volatile. It changes a lot during early ontogeny, and then it sort of settles down into essentially the adult pattern at some point, in relatively late in ontogeny. But I think we don't have enough sampling to know, is that a normal pattern? It's, most of the sampling that we have is in mammals. So is that unusual? Is that related to, does that vary based on differences in, reproductive strategy? What is the actual, ramifications of that? So you could imagine that if you have taxa that are kind of under, so one of the things I thought was an interesting question, right, is if you have species that have functional pressures at different points in ontogeny where they have very different patterns of integration from their adult state, you might see different kinds of effects on the ultimate variation in that organism compared to things that don't have those sorts of pressures at periods of really high overall integration. But I think, at least some of the suggestions are that integration might actually, early ontogeny is very integrated, and then you get more modular as you become an adult. but again, you get a lot of kind of repatterning through ontogeny. So I think ontogenic integration is super interesting and definitely very consequential. But I think that, I think there's a lot that we don't know about it because we just don't have that much data on it. Totally possible to capture that kind of data though, which just hasn't really been done that much yet.
Okay, so Anjali, well, I don't see any more hands, so I will ask mine. Well, I have plenty, but we used to reduce to two. One for you give an advice to the students that are just now watching you, because I receive many questions like, it is this kind of figures are very amazing. They're so hard to do in that. It's difficult in terms of processing. So it is difficult to digitize all the digitalization. So kind of software that you do. You did using. I see you using Morpho for the kind of analysis. But
Yeah.
for example, there's Alpaca, also new software from SliceMorph that do some kind of volume and declaration. It's kind of something similar. How do you recommend to students to do that?
Oh, it depends a bit on, I guess, what your question is, right? Because if you, that's, I guess, a standard cop out, but it depends on whether you want to look at individual regions or if you want to look at the shape of an entire structure, in which case maybe you could use pseudo landmark approaches. And whether you want landmarks versus semi-landmarks will also depend on kind of the scale of analysis that you're doing and the structure that you're trying to study. I think the really good news about all this is it's actually a lot easier to do this now than before because there are so many with like the Overt project and Morphosource and multiple other, like Phenom10K and other databases coming out there. The access to having 3D data has gotten, immense. When I started this project on the vertebrate skull 6 years ago now, there was nothing like that available. But now, it's actually entirely possible to gather big 3D data sets without actually having to travel very much. And so that makes things a lot easier. And the other part of that is alongside having open data, there is really great access to open software or free software now. So, we use Aviso in my lab just because we've been using it for a long time. But if I was starting now, I would just use Slicer because it's free and it works well. And so you don't really need this really expensive software to do these things anymore. I haven't used Alpaca myself yet. I think, but I think, a few people in my lab have tested it out and said it looks like a good tool that is probably fairly similar to the way we do our approach. It is true that these are difficult methods though, and that they are quite time consuming. Slicer, sorry, I just saw that chat what the name of the free software is. Slicer or Slicer Morph. Slicer is the 3D reconstruction part of it. But yeah, so they are time consuming. They are hard to do. There's a lot of tweaking, but there are a lot of people that will also be willing to help you. So, certainly, members of my lab do spend a lot of time helping other people when they have questions. We put out a paper really describing what we do in painstaking detail that Carla Bardua led. We try to include like, okay, this is how it'll screw up here. So if it screws up this way, you can deal with it in these different ways. If you deal with it these different ways, these are the different things that will happen. Because there is
fantastic.
Yeah. But yeah, so there's lots of good options, and there's a lot of people that are willing to help you navigate how to use them. So I would say just dive in. That'd be nice.
Yeah, definitely. And definitely there's more option now than 6-7 years ago. The option to go and surf in different scan is easy now. So okay, so the student just have to go and try to do some nice stuff. So now
and don't be afraid to ask for help ever.
Hey, cool. And now in terms of the talk, I have a question regarding to all the what you seem to your talk are regarding to vertebrate and evolution of vertebrate, integration of this big disparity that you see in the evolution of vertebrate. But when you go into the invertebrate, there's something that for example, flies. In terms of integration, you see, let's go to one sample like Drosophila, the wings are very, very similar between, there are more than, I don't know, 1,000 species in one genus, but
Right.
where all the wings are kind of very similar. So in terms of integration, so having a high level of integration, because the wings have high levels of integration, and is it related to the very stable morphology, or is the integration reduce the evolution of disparity, like you did in your simulation? Are something related to high levels of integration in one structure with the evolution of disparity?
So what do you mean? Do you mean that because they have so much species diversity, but not a lot of morphological diversity in that one structure? Yep. And if that's related to the structure being highly integrated, yeah, that's entirely possible. That would make a lot of sense. I mean, I think that would definitely be my null hypothesis for why that structure is so, so stable in that sense. Especially if other aspects are really variable.
Because when you talk about modularity, so related with diversity, so biological diversity are related with more modularity, so it could be the contrary. So having high levels of integration in some structures could be no more disparity or no more evolution. I don't know. I just thinking.
No, I think that could make sense. But I would also distinguish, taxonomic diversity from morphological diversity, right? So you can have massive amounts of taxonomic diversity with almost no morphological diversity. Yep. And whether that's related to those taxa being more integrated than others, I'm not entirely sure actually if that has to be the case. I think it could actually go either way. But I would say that my expectation is that it would probably be because of that. But you could see things happen the other way also. If it's entirely. Because sometimes the pattern is not just because of the intrinsic constraints, it's because of environmental interactions, right? And so, so you can't be 100% sure that disparity hasn't evolved because of a lack of capacity to evolve the disparity, as opposed to competition and things like that.
Exactly.
And you also have to, one of the other things to worry about is the sampling and whether there is a lot more diversity in the extinct. And the full, played sample of extinct things that we're not representing. But there's only so much that we can control those things.
Okay, I think there's no more questions. So I have one announcement to do before. Well, first I have to thank you very much to you, Anjali, for your amazing talks. For sure will be inspiring for all the students for Latin America and also for the Chilean Society. Give you the big thanks for your time to be with us. I have to give an announce in Spanish for all. So para todos, bueno, estamos muy contentos con la Sociedad Chilena de Evolución de que estén todos presentes. Queremos dar un anuncio en términos de un congreso que estábamos planificando con las diferentes sociedades de evolución en Latinoamérica. Voy a compartir y si es que están todos viendo, tenemos ya fecha para el primer Congreso Latinoamericano de Evolución, en la cual se juntan las diferentes sociedades evolutivas en Latinoamérica, que se realizará entre el 30 y el 3 de diciembre. Las inscripciones, registros, se van a entregar por los diferentes medios de comunicación, Facebook, Twitter, Instagram, de las diferentes sociedades de evolución en Latinoamérica, la próxima semana, en donde vamos a conocer ya las fechas tentativas de entrega de resúmenes. Así que para que estén pendientes con esto. Y con esto doy por finalizada la charla. Thank you very much, Anjali, for being with us. And thank you for everybody to be with us today. And see you in the next seminar of the Chilean Society of Evolutionary Biology.
Thanks, everyone.
Gracias.